Hello, everyone. My name is Weronika, and I work with Sarah. And I'm very happy to present right after her because she already gave you a beautiful introduction to our pupil-based biofeedback that we use in our lab, and already went through the many protocols that we use to validate this pupil biofeedback. Now I want to mostly talk to you about arousal, self-regulation, and stress response. And a little disclaimer, this is more like a planned study that we are working on. So I will show you some first insights into our planning mostly and our testing of the protocol. So I knew that's gonna happen. Thank you. Yeah, so you're all stressed from time to time, for example, because we see a monster in the room, as Lucy explained to you, but also in the situations like this one, for example, when we need to give a talk and we can feel that our heart starts to beat a bit faster, our mouth maybe gets dry, our hands are potentially shaky. What we don't see is that our pupil increases, our blood pressure potentially also increases, and what follows the stressful situation is the release of cortisol in our body. Now, Sarah already mentioned lots about it, so I will just go quickly through this part. There is a small brainstem nucleus called locus ceruleus that is partially responsible for those changes. And LC is the main source of noradrenaline in the human brain, and it releases noradrenaline to modulate various functions, and is involved in modulation of various functions. For example, it's shown to modulate the arousal level, but it's also involved in bodily changes that we collectively call stress response. And interestingly, it is also involved in pupil control, which is particularly important for us because pupil size can then serve us as an indirect readout of the LC activity, as Sarah nicely pointed out. And we demonstrated in our lab that we can use this link between pupil size and LC activity in a pupil-based biofeedback protocol where people can train how to up and down regulate their pupil size, and these changes were associated with the changes in the LC activity. Now, knowing all about the physiological underlying mechanism that Sarah pointed out to you, we wanted to check whether we can use it for something a bit more applied this time. So we want to check whether this pupil-based biofeedback can serve as a method to modulate the stress response. So we plan, or our idea is to ask participants that are already trained in the pupil-based biofeedback to apply their mental strategies to control their pupil size. That would then hopefully change the activity in the locus ceruleus, and by that, whether we can potentially modulate the stress response that is known to be driven by the noradrenaline as well. But to modulate the stress, we first need to induce stress, and we decided to use the protocol for stress induction that is called socially evaluated cold pressor test, in which we ask participants to put their hand in the ice cold water while being observed by an experimenter, and the online recording of their face is displayed on the screen right in front of them. So that's not particularly pleasant setup, as you can imagine. But what makes it even more difficult and hopefully also stressful is the unpredictability and lack of control because we don't tell them how long they need to keep the hand in the water. And such intervention is shown in the literature to increase variable parameters of the stress response, like pupil size, heart rate, blood pressure, cortisol level after the intervention, and also it's reflected in some self-reports like how stressful, painful, unpleasant it was, and also in the anxiety questionnaires. So, yes, it looked a little bit different before, but the bottom line here is that the others will also be like that. So the bottom line is that what we can see, for example, in the anxiety questionnaire, is that people show higher scores in anxiety measures measured by style questionnaire, post versus pre intervention. And we could also see an increase in the blood pressure during the intervention, heart rate, and pupil size. Luckily, this one did not lose all the qualities that I hoped for. Following the intervention, we could also observe increased cortisol release. And as said, you can see very small sample size. Here we really wanted to just validate whether our stress intervention actually induces stress responses. But now, of course, the question remains whether we can modulate this. And our idea is, again, we will ask participants to downregulate in this case. So we would have two different sessions, one when people downregulate their pupil size, so using their mental strategies, and in the other control condition, we will just ask them to count backwards to keep some active state of the brain, but without applying any strategies. And we would combine it with our socially evaluated cold pressor test, and afterwards, we hope to see maybe some potential changes in the parameters of the stress response, reflected in decreased pupil size, decreased heart rate, increased heart rate variability, decreased blood pressure, cortisol, and some parameters measured as self-reports. And I will just show you first very small insights into, again, our protocol tested with these two sessions, but it all happened before we really started a full-blown study. And to let you understand the charts that I'm going to show you in a second. So people took part in these two sessions, and we used our intervention in these two sessions in the two conditions. And what you will see later on the slides is that on the x-axis, there is a pupil modulation, a control minus down. So basically the higher numbers you see on this scale, that means that people downregulated better. And also for our outcomes on the other scale, it's also our control condition minus the downregulation condition where we expect decreases. So basically, the higher the numbers, the bigger effect we could see in people downregulating. So basically, the downregulation brought the values lower, which are indicated here as higher values. I hope that makes sense for you. So what we want to see is rather the tendency to the upper corner. So first that we can definitely see is here on an example from the self-reports. And again, as I said, just a very small sample to basically test our protocol. The first important thing to notice is that people really vary in how well they downregulate. So we have people who can really perfectly downregulate and people who struggle a little bit more. So the first important message is the variability among the method, the modulation basically. And as said here, no clear trends in the self-reports of anxiety at this level of the setup test. But for example, already in the pupil diameter, we can, of course, not talk about any clear correlations here, but there is some tendency showing that people who can better downregulate could also bring their pupil size basically lower during the stress intervention. And the same holds for blood pressure. So basically, people had potentially lower blood pressure in the stress test when they were applying downregulation. Interestingly, also, we could see something similar direction for the cortisol, which we were very happy to see at this point. So that the cortisol release was slightly lower in the people who downregulated. No clear results for now with this sample. For heart rate, and also kind of similar tendency for heart rate variability. But as said, what I'm showing you here mostly is we basically validated whether our setup makes sense in the lab. And I came here also to get as much feedback from you as we potentially can, because we are, as said, just starting the study soon. So we are still clarifying our protocol and thinking about potential measures that or analysis basically that we can apply to the signals that we collect. So any feedback would be very welcome. And as conclusions, we could see that we could reliably induce physiological stress responses in our participants with our intervention. We have our first insights into the effects of self-regulation on the stress response. And of course, that awaits further validation and the full study. And thank you for your attention.
Okay, questions?
So thank you. The results, even if are only on 6 subjects, looks quite, positive in a sense. My question is about your manipulation. You decide to add to the cold pressure test, which is one of the tests used both to measure pain perception, but also to induce stress, this social, factor. Do you have, are you planning to include measure to try to disambiguate if your downregulation, pupil-based feedback actually act in, downregulate the social stress or the perception of pain? Because this could be, useful to understand which...
No, totally valid question. Thank you for that. We were actually also thinking about it a little bit. We haven't fully decided how to disentangle. The reason why we decided to go for the socially evaluated version was that one of our collaborators, she actually works a lot in the stress domain, and there was one of the advice that she gave that the results are basically more robust if we have this social component. It also is quite clear from the literature that the effects are just bigger when the pain basically is combined with this aspect. So that's why we went for it. We also wanted to of course, it's still not very ecologically valid, but we thought for our how we understand future potential stress management with our pupil regulation, typically people would hopefully profit from it in social situations that we are exposed to, and they're more stressful for us in daily life a bit more often than these physical stressors. So that's why we wanted to go in this direction, and in the future we may even go to other, even more social or cognitive versions of the stresses. But answering your question, I mean, the only thing that we can do at this point, we have the visual analog scale, for example, at the end, where we ask how stressful, painful, and unpleasant it was, and then on top of that, anxiety questionnaires before and after.
Social measure of their, tendency to be, I don't know, stressed by social situation.
That's what you mean.
I don't know.
Yeah,
there is this anxiety sensitivity questionnaire in which you evaluate your, let's say, physical sensation, and you are catastrophizing
exactly.
it
Yeah,
with respect to this.
We have set of questionnaires before participants even start the whole procedure, and one of those, it's this catastrophizing of the pain sensation, and also all sets of potentially of questionnaires measuring different exposure to stress throughout life. So we could also maybe divide people into those that were a bit more exposed versus the others. The one that you just mentioned about specifically social, I will go back to you afterwards to get the name again, because it would be maybe really useful to include that as well. But yeah, that's actually a good idea to collect the information before and then test them separately. But thank you so much.
Sure.